it's recorded. So you can share screen. That's what I want to do. Okay, so we're going to have a look at proteins today. So sort of starting off with the primary structures of proteins and building up to the quaternary structures of proteins. Then next week or later on in this week, I can't remember when I've put it down to do it now, we'll start to look at specific proteins like enzymes and DNA and RNA. But proteins is quite a big topic in itself. So it builds on this polymers and monomers topic again. So it starts off quite nice and easy. We're starting off with the monomers, building up to the polymers, and then proteins are quite complex. They're quite cool. So why is my slides not moving? So we're going to look at the structure of the amino acid to start off with. So that is going to be our monomer. We'll then look at amino acids joining together to make polypeptides, which will then be our polymer, our primary structure, and then look at the 4 structures of proteins. So how we then get this 3D shape. So when we've been talking about carbohydrates and lipids, and they don't have this same 3D structure that a protein does. So proteins have these really complicated structures to do with different bonding that takes place as well. So we'll look at them and look at the test for proteins. Test for proteins, again, it's not changed since GCSE, but we have to know that as well. So proteins. Proteins are really, really important. So they make up 50% of a dry cell's mass. So if you dried out a cell, got rid of all the water that was inside of it, 50% of what was left would be proteins. And they're really, really important because basically they make up the vast majority of the really important parts of our body. So our cell membrane is made up of proteins. Obviously, its cell membrane is made up of lipids and proteins, but they're in there with the cell membrane. Our hemoglobin in red blood cells are proteins. Antibodies, which really help with our immune system and start to help us fight off different infections, are proteins. Enzymes, which come up all the time in chemistry and biology and have a huge array of jobs, are just proteins. Keratin in your skin and hair are proteins. Collagen is proteins. So they make up so many different things, and we often don't call them proteins when we're talking about them. So occasionally we'll talk about proteins, and often when we talk about proteins, we're talking about the food proteins. But all these things I've just mentioned are also proteins as well. So we often never really refer to enzymes as being proteins or DNA as being protein. So it's sort of one of the things we have to just keep checking back in our head and going, oh yeah, no, they're made from amino acids, so they're proteins. So what are proteins made from? Proteins are basically just really, really long chains of amino acids. So they are dipeptide, really similar to when we talked about disaccharides with carbohydrates. So these are dipeptide when 2 monomers join. So 2 amino acids join together, we have a dipeptide. And then when we have 2 or more amino acids joined together, we get a polypeptide. So proteins are made of 1 or more polypeptide chains. So this is where we start to get a bit more complex than we did with carbohydrates and with lipids, because here we're starting to say proteins are made of 1 or more polypeptide chain. So when we're getting into that tertiary and quaternary structure, we're actually getting multiple polypeptide chains and putting them together. So we have lots of intermolecular forces at play. But they all have the same general structure, which is quite nice. So amino acids, this is our basic structure, of an amino acid. So I'll talk it through with you and sort of explain the different parts. Again, this is something that you have to know is the generalized structure of an amino acid. So we have this end of our amino acid. Let's draw a line from here. I'm going to actually change where I've got my pen because that is not working as well as I wanted it to. That's better. Right. So this end we call the N-terminal. Literally, it's got its name because of the nitrogen in the amino acid. So this is our N-terminal, and this is an amine group. Some call it an amino group, an amine group. as we go more into chemistry, you'll get more used to things like amine groups. So this is our amine group that's on our amino acid. So amino acid, amine, the name amine literally just comes from the amino part. And then we have an acid on the side. And so we have a carboxyl group, a carboxylic acid. So this is our carboxyl group here. And this is what we call our C terminal. C, because it's our carboxyl group. So we have our carboxyl group at our C terminal, we have our amine group at our N terminal, and then we have our side chain. So we have our R groups, our variable groups. And these are the only things that change on our amino acids. So these are our variable groups, also sometimes called a side chain. So we have 20 amino acids in total. And the main structure, the main generalized structure of amino acids stays exactly the same every time. The only bit that changes is this R group. For example, let's get the next page. Rid of annotation. Did not realize I was going to do that. Okay, so clear ordering. There we go. So amino acids, we have 20 of them occurring naturally. We have 20 naturally occurring amino acids. 10 of these we have to obtain via our diet in animals. These are what we call our essential amino acids. So these are the ones that we will get through specific foods. 10 of them we can make in our own body, but 10 of them we have to obtain via diet. So it's why when people go on all the time about having a really, really rich and varied diet, making sure that we're eating fruit and vegetables that have got vitamins and minerals in, they're really important because that is where we get lots of amino acids from. As we've just seen, they all have the same structure and they just have different R groups, which we have here. We've got the R group here, so it has a different R group. And glycine is our simplest amino acid. The R group is just a single hydrogen atom. So glycine, and I'm just going to draw you a glycine on really quickly, would look like this. So we've got our N terminal over here. So we've got our nitrogen with 2 hydrogens coming off it to make our amine group. Carbon in the middle, go into another carbon, make our carboxyl group over here. And then what would normally be our R variable group for glycine is just a H. So that is just a hydrogen. So that makes it glycine. We can get loads more complicated. Just using glycine as an example, as it is our simplest amino acid, but we don't have to learn them. We don't have to know the whole list of the 20 amino acids. So don't worry about learning all of them. You don't need to. We need to know this basic structure of amino acid, but we don't need to know all 20. I'll show you some of the 20, just because sometimes they show them in exams and just so you're more familiar with them, but actually you don't need to know all 20. Oh, and there's that basic structure. That is not helpful. Okay, let me get rid of my writing again, clear up drawings. So we have a primary structure formation of a dipeptide. And what we mean by this is protein is made up of loads of amino acids joined together. Got amino acids, we're going to join them together. When we join 2 amino acids together, we get a dipeptide, same as we join 2 monosaccharides together and we get a disaccharide. So we have this prefix in here again of di, meaning 2. And peptide is part of our polypeptide chain. So it's a 2-chained, 2-part chain. So let's get this open. So here is 2 amino acids. Both say R1 and R2. R1 and R2, they could literally be anything. They're just our side chains. They could both be exactly the same. They could both be different. It's just sort of to prove a point that it doesn't actually matter which 2 amino acids are joining. The way that they join is the same. Exactly the same as we were saying last week is these are going to join using a condensation reaction. So we have 2 amino acids. The OH from one carboxyl group of the first amino acid and the hydrogen from the amine group of the second amino acid are going to join together. They're going to go and form water. So we're going to form H2O. So our condensation reaction is going to happen. So H2O is going to come off, and we get this bonding happening instead. So this, we start getting a straight bond from our carbon straight to our nitrogen, and we've lost our water. So that is our condensation reaction. This is to form a dipeptide. This area here, we have made down here something called a dipeptide bond, or a peptide bond. You can see it written 2 ways in books. It's really annoying. People interchange it, which is slightly not helpful at all. But it is a peptide bond. I prefer just to call it a peptide bond. But yes, this is a peptide bond here. So whenever we're making peptides, joining them together, whether it's a polypeptide, a dipeptide, we make a peptide bond. It is a type of covalent bond. Is a type of covalent bond, but we call these specifically a peptide bond, and that is a joining of the carbon from one amino acid to the nitrogen of the other amino acid. So we get a peptide, a dipeptide. And basically, this is our primary structure of a protein. And primary structure of protein is just amino acid joint to amino acid joint to amino acid. Obviously, we have 20 different amino acids. So this could be, for example, glycine. This one could be another glycine. We could have thymine in there. So we could have loads and loads of different ones all joining together. And it doesn't really matter too much which one's where. So clear all drawings. Let's move this one along. So our amino acid structure, we've got our R group again. It's just a different way of looking at it. So this amino acid, we're just having a quick look at the R group on this one, has a carboxylic acid in the R group. Therefore, it has 2 acid groups overall and 1 base group. So overall, this amino acid is an acidic molecule. This isn't something that we have to worry about too much in biology, but I've added it on with you covering chemistry as well, because it starts to get quite interesting. Whereas, for example, this amino acid has an amino group in the R group. So therefore, it's got 2 base groups and only 1 acid group. So overall, this amino acid is a basic molecule. So whilst officially they're all called amino acids, some are more acidic than others, some are more basic than others. So we do have this balancing act. So it's just that sort of a bit of extra knowledge that's quite interesting, I find, about them. So don't know why I've got this one on there again. Here we go. So our primary structure of a protein is just its polypeptide chain. These 3-letter shorthands of the names of the polypeptide, of the amino acids, sorry, you don't have to know them. The more you sort of read, the more you sort of pick them up, but you don't actually have to know them for the exam. So we've got histone, glycine, serine, alanine, valine, lysine, sort of lots of them end in "ine", to be fair. And so the primary structure is literally, primary structure of proteins is just the chain of amino acids, the order of these amino acids. So the fact that we're going 1, 2, 3, 4, 5, and the order that they're in. So on one end of a chain, there will be an amine group, and on the other end, there'll be a carboxylic group. So what we mean is, when we see that original drawing of our amino acids, where we've got, I need to change my pen if I'm going to do that. We've got this going on. No, that's not what I want to do. Right, here we go. C double bond O, OH, HR. So what we mean by that is, on this end, coming off this histone, there would actually be an amine group that's not bonded to anything. And coming off of valine at the end, there would be a carbon. We'd have our carbon coming off here, so that'd be our carboxyl group, carboxylic acid group coming off the end. So they're just the loose ends that are there, and that's all that means. Nothing too fancy going on. So that's our primary structure. Nothing too complicated there. Another way of looking at primary structure is amino acid, amino acid, amino acid, all joined together doing condensation reactions. So we've lost lots of water to join them together. Then we get on to our secondary structure. Our secondary protein structure can have two different forms, and it can come either in an alpha helix or a beta pleated sheet. And this is all to do with hydrogen bonding that's going on between them. And so we get this specific folding. So we'll look at them in a little bit more detail now. So secondary structure, we can either have an alpha helix or a beta pleated sheet. And this is all about the interactions and the bonding here to start to make this 3D structure. So rather than just being a flat polypeptide chain now, we're starting to get folding. We're starting to, if we think back to when we were talking about storage molecules like cellulose and amylose and amylopectin, those coils, those spirals, because of the intermolecular forces and the bonds in play, this is what we're starting to get here. So first example is an alpha helix. Alpha helix you'll start to become quite familiar with because it will start to come up a lot because this is part of how our DNA is shaped. So alpha helix makes this really pretty helical shape we have down here. There isn't, and these pictures always annoy me, there isn't actually a central column to them. They always draw them in here and I don't know why. Basically what happens is alpha helix is brought about by the attraction between our carbon double bond oxygen on the end of your carboxyl group of one amino acid and the NH group, so the amine group of the amino acid 4 places along. So if we counted them on the chain, let's try and number them. So we would have number 1, number 2, 3, 4. Yeah, no, that isn't working. 1, 2, 3, 4. Don't trust a picture is what I'm going to say now. 1, 2, 3. 1, 2. There's an answer for you. Don't trust pictures where it's saying what they are. But it should be the carboxyl group. So here we go. The interactions between that and the amine group 4 places along the chain. So what happens is hydrogen bonds are formed here. So we've got our H bonds. And what happens is it causes the polypeptide chain to coil up into this helix. So as you can see, you have these reactions, these hydrogen bonds forming, which causes this helical structure to form. Another way of showing it is we also have our beta pleated sheets, but we will have a little look at our alpha helix again as well. So we have our amino acids, our peptide bonds, and these are hydrogen bonds forming. These ones are showing them between the 1 and 4 a little bit better. So we have amino acid 1 here, 2, 3, 4, and we've got the bonds forming between them. 1, 2, 3, 4, and the bonds forming between them. So you can see between number 1 and number 4 a lot clearer on this image. So that's amino acid 1, amino acid 4. And if we started the numbers again, but it's 4 places along. So 1, 2, 3, 4. If we count them up, that would be our 5th one. Yeah, that's just how we would go. So 4 places along is where they're going to be, and that would be our hydrogen bonds before them, and that's what forms these helical shapes between them, this helical, really nice helical structure. It's those hydrogen bonds between the carboxylic acid group and the amine group, and this is our alpha helix. We also get our beta pleated sheets. Beta pleated sheets are a lot much more of a looser shape than alpha helix. The alpha helix is this very defined coil shape. Beta pleated sheets are a bit different. So it's working on hydrogen bonds again, but this time what is happening is, if you have a look, is they're lying flatter, and basically we've got a polypeptide chain that then forms here the hydrogen bonding across. Now the R groups that we talked about, these extra additional groups, these bits that say which one of these 20 amino acids it is. So here we go, we've got our N, our H, C, C, OH, H, and our R. These R groups determine whether we're going to make one of those alpha helixes or a beta pleated sheet. And we'll look at beta pleated in a bit more detail now. So this is going to go through our beta pleated sheet. Again, we're starting off with the same polypeptide formation like we do the alpha helix, but this time we form a beta pleated sheet just because of where the bonding forms this time. So this time the hydrogen bonds form this way, whereas before they were going between 1 and 4, so you'd have 1 and the fourth one along, it would make a hydrogen bond. This doesn't work the same way. This way we're going straight down each time on these patterns. So basically we've got a repeat of the patterns here and it's going through. So if you look, if we numbered this one, this is when I get 4. 1, 2, 3, 4, 5, 6, 7. We've got 1, 2, 3, 4, 5, 6, 7. We've got them forming below each other each time, so it's the same ones going each time. So if we call that one 7 and that one 6, we're going to form again this way. So there are hydrogen bonds forming there. Clear that. Hydrogen bonds forming, and that makes a beta pleated sheet. And what actually happens is we have these little end parts where it loops together. So now this will make a lot more sense. Let's number them. So we've got 1, 2, 3, 11, 12, 13, 14. We've got 1, 2, 3, 4, 5, 6, 7, 8, 9. No hydrogen bond happens on these loops around the side, and then 10 forms here. Then 11, 12, 13, 14, 15, 16, and again, no hydrogen bonds happening on these forks at the end again here. So beta pleated sheets and alpha helixes, they are the two different types of secondary structure we can get. Now, they are not just solo. And what I mean by that is a polypeptide chain of amino acids can coil to make an alpha helix. They can make a beta pleated sheet, but actually in reality, what they often make is they make both. So what we have here on this image, we've got the red tubes are symbolizing alpha helixes, and the purple corrugated sheets are our beta pleated sheets. And we have these things called an amorphous region. So what basically actually happens in reality is our protein is actually made up of different sections. Some of the sections will be alpha helix, some of it will be beta pleated, some of it might all just be alpha helix. So for example, when we're talking about DNA, it is all alpha helix, but we have different sections, and we have these amorphous regions, which just means it doesn't have any defined specific shape, and that just means that the R groups in those areas aren't interacting in a certain way, which means that we're just going to form the sheets in these areas and we leave these gaps. And then we go on to our tertiary structure. So this is why it's really, really important to remember that whilst our secondary structure is made up of alpha helix and beta pleated sheets, it's the and that's important. So it can be just alpha helix, it could be beta pleated sheet, or it could be alpha helix and beta pleated sheets. And that's just the thing just sort of keep in mind about the secondary structure. It's not this or that. It could be an and as well. So then we go on to our tertiary structure, and this is where it gets quite interesting. This is where we start to get them 3D shapes coming from. So this tertiary structure folds up to start to form this precise 3D structure. And this is where those R groups that we've already talked about once, deciding if it's an alpha helix or a beta pleated, come into play even more. And so when I was saying that some of them cause the amino acid to be more acidic or some of the amino acid to be more basic, that really plays a part in this folding up to make the tertiary structure. So the alpha helix and beta pleated sheets are folded, or they can be coiled around a three-dimensional shape. And what starts to happen is we start to get other types of bonds coming into play now. Up until now, we've literally just been talking about hydrogen bonds. Now we're going to start getting some extra ones, and these form between the amino acids to stabilize them, and specifically the side chains. So when we've said that one amino acid could be more acidic, one could be more basic, this is what's sort of going to create this stability. So the shape is very, very precise and specific to the function of a polypeptide. So for example, DNA is an alpha helix. Well, it's a double alpha helix, so we actually have the two. When we're looking at things like enzymes, these are really, really precise and specific shapes. Each enzyme has to have a specific shape for its active site. If we're talking about receptor proteins, transporter proteins, they're all really, really dependent on the size. It's that lock and key enzyme theory basically coming into play. There's so much about proteins here. So this shape has to really, really be right. And that's all determined by the order of those amino acids from its primary structure. So this is quite a good question. It's one that comes up a lot. So what happens to the function of a protein if the amino acid sequence changes through mutation? Well, if we go back and we think that we had amino acids coming along here, let's draw these on a bit better. So we've got amino acid 1 joint to amino acid 2, joint to amino acid 1, joint to amino acid 3. They will have their own specific folding process that happens. It'll make its own precise shape or do its intermolecular forces and bonds. If for some reason we had a mutation and amino acid 2 becomes amino acid 3, the rest stays the same, the shape will change. The shape of the protein will change, which then will lead to the function of the protein potentially not working. It depends how big a difference this change causes. If you think that we've just said they're really, really highly specific, if we think about enzymes, the active sites have to really, really stay the same. They have to be really, really specific. If one thing changes, if just the folding in 3 changes, just swapping from 2 to 3 might have made this be an alpha helix, and then this one becomes a beta pleated. I'm sort of simplifying it there, but sort of make a point that we can then change it completely, which then just makes the whole function of that protein change or actually not work at all. So mutations in proteins are really, really major. So let's have a look. Our tertiary structure. The tertiary structure is when our protein is going to coil up to form those precise 3D shapes. These include alpha helixes and our beta pleated sheets, and the shape it forms depends, like we've already said, upon that sequence of amino acids, as it's the interactions that are held between them. So these are sort of, this is our tertiary structure over here. So this area would be our tertiary structure, and this is showing that some areas are made up of alpha helix and some areas are made up of beta pleated. So proteins are 3D. They become 3D because of 4 different types of side chain interactions, which we're going to look at in a little bit more detail. Hydrogen bonds, again, are going to come into play, like they already have done to form those alpha helix and those beta pleated sheets. We also have disulfide bonds, ionic bonds, and hydrophobic interactions. And these are all very much dependent on the R groups. And I'll use some examples of some of the R groups as well. So, hydrogen bonds. So some R groups are polar, And so the hydrogen bonds may form between them. So we'll have a little bit of a talk about polar and non-polar molecules. So our R groups, 3D shapes are formed by our R groups. That's the most important thing. And our R groups are polar when our electron sharing is not even. So a molecule that we'll do loads about at some point is water. So H2O, let's draw our oxygen. You'll start to see it's always drawn like this. H2O is a polar molecule. Polar molecules occur when our electron sharing isn't even. So, for example, we have 6 electrons on our oxygen, 1 on each hydrogen, and it creates this polar molecule. Okay, so it's not being shared fully equally. So it creates these interactions. So polar groups, so our groups are polar when electron sharing is not equal. So I'm just going to put E sharing is not equal. What happens is polar R groups attract other polar molecules. It's a bit like when we say positive attracts negative. So polar groups attract other polar groups. So for example, water, here we go, we've got one water molecule here. Water likes to attract water. So water, when we start to look at transpiration and evaporation, water has this potential, because it's polar, to attract other water molecules to it. Okay. So water, polar molecules attract other polar molecules. So water molecules to each other, we would say they're hydrophilic. They want to be near each other. They are drawing each other closer together. So these are hydrophilic, water molecules are. So polar molecules are hydrophilic. Non-polar molecules are non-polar R groups, sorry. So polar R, let's put this here, polar R equals hydrophilic. Non-polar R groups, so these are going to be R groups that have equal sharing of their electrons. Non-polar R groups are going to be hydrophobic. Means that you're not going to want to be near water. So we can start to see that actually we're getting a push and pull here. Some are going to want to be closer to water, some are going to want to be further away from water. So we're getting these interactions, these intermolecular forces. This is going to start forming a shape. So non-polar R groups, what's going to happen is they're going to face the inside of a molecule because they don't want to be near the water. So our non-polar R groups are going to face the inside of the molecule, excluding the water from the center, and we're going to have our polar R groups on the outside. So if, for example, I'm going to draw it badly here, these are our non-polar R groups, and they're drawn in black just for the sake of it, and then our polar R groups are going to be on the outside. It's really badly drawn, but you're sort of getting the idea that basically because they're hydrophobic, they want to go in the middle. It's a bit like when we talked about the lipids and they're making micelles or phospholipids and they're making the bilayer. The tails are hydrophobic, so they go into the middle. The heads are hydrophilic, so they're on the outside. What happens is the chemical bonds start to form between the R groups, which are close to each other. So we get this clumping together of hydrophilic and hydrophobic R groups, and then the R groups will start to interact with each other. So some R groups are polar, and these polar R groups that we've just been talking about, hydrogen bonds start to form between them. So some R groups that are polar are hydrophilic ones, the ones on the outside, the polar, so they're going to start possibly making hydrogen bonds between them. Then if my computer would move on, here we go. we can have, I can't see what I've typed at the top, but never mind. We can start to get disulfide bonds as well. So these are quite interesting. Let's move this a second. Oops, no. So we can have disulfide bonds. For example, we're going to use cysteine as an example, which is another type of amino acid. I couldn't see which one we were using then. Okay, so cysteine, we have our R group here, which is S and H. So we've got sulfur and hydrogen. What happens here is between two cysteine molecules, we get this disulfide interaction, this disulfide bond taking place. So here it is here. And that's between the two sulfur molecules. So oxidation has to take place. Oxidation, if you remember back to GCSE, oil rig, oxidation is loss, reduction is gain. Oxidation is going to happen. Our two sulfurs, join together to make a sulfide bond. So it's disulfide bridge, as we often call them. So we have a cross-linked pair of chains, and the link is a disulfide bridge, so this SS bridge. These can be broken by reducing agents. This is something we'll come back to later, but it's quite good to sort of remember. So cysteine, two covalent disulfide bonds. These are stronger than hydrogen bonds, but for example, a reducing agent in this situation, we could talk about something like pH. They are very, very, very sensitive to pH. So a change in the pH slightly, and these disulfide bonds, these disulfide bridges can break. Then we have another image. I'm going to show those bonds in a protein again. So here's a protein. It's in its tertiary structure. It's all squiggled together. And here's some examples. So here we've got an example of a hydrogen bond. That's involved in all levels of the structure, apart from the primary structure. So going all the way up, we have these hydrogen bonds involved. So between the carboxylic acid group and the amine group. Then we have these hydrophobic interactions happening. So these are happening between our non-polar sections of our protein. So these are going to be our non-polar R groups, and they're going to have hydrophobic interactions together to make this area that stops any water being passable. Here's one of those disulfide bridges that we just spoke about. So these are one of our strongest and most important type of bonds in proteins, and these happen between our cysteine amino acids. And the last one is ionic bonds, which apparently is not going to appear. Hydrogen disulfide, ionic, here we go. I don't know what's happened to my slides here. Right, let's go past hydrogen bonds again. We've already spoken about hydrogen bonds. Okay, let's quickly do hydrogen bonds again. So, quick recap again, hydrogen bonds, they're going to occur between our polar molecules. Our polar molecules have slight charges on them due to the way the atoms are made from the shared electrons. So they have an uneven share of electrons, which makes them polar, which means that hydrogen bonds are going to form. This means that the charge is going to be either slightly positive or slightly negative. As we know, opposite charges attract, and this attraction forms a hydrogen bond. So in proteins, the hydrogen from the amine group tends to be slightly positive, whereas the oxygen in the carboxylic acid part tends to be slightly negative. Therefore, they attract. So here we go. Here we have an example. Have our carboxyl group, we have our amine group. We've got these shared electrons spend longer at these atoms, forming a slightly negative charge. And then our hydrogen bond forms here. High temperatures and altered pH can split these bonds. These are the weakest bonds that we're going to talk about here. Then we have, oh, I don't know why I've got the hydrogen bonding. We do not need to do this again. Our disulfide bonding, only between cysteine amino acids. It's removed by oxidation, and we get this disulfide bridge forming. So cysteine again, disulfide bond, type of covalent bond, and disulfide bonds can be split by our reducing agents. Ionic bonds, where I wanted to go. So ionic bonds, exactly the same as ionic bonding in GCSE. These are attractions between atoms that have been ionized, so they've either lost or gained electrons to become positively or negatively charged. What happens here, oops, yes, I did want this one, is we have our bond that bonds it to the rest of the molecule, or bond to the other rest of the molecule. So we've got our amines going on and our carboxyl group. So we have our acidic group and our basic group again. Now our ionic bond forms, as you can see, in a very similar place to where our hydrogen bonds form. Ionic bonds can be split by changing the pH. They are a lot stronger than our hydrogen bonds. And those hydrophobic interactions that we've already spoken about. Hydrophobic molecules are those that don't like water. Therefore, they don't mix with water because they are non-polar. Water is a polar molecule, so it is hydrophilic. Amino acids that have non-polar R groups can form hydrophobic interactions with other non-polar R groups. And this occurs because they cluster together away from the surrounding water. Here we go. We've got a picture of them on here, and I'm just going to quickly add on what we've got. So here we have an example of one of those disulfide bridges. Then we have an example of our hydrogen bonding. We have an example here of our hydrophobic interactions, and then down here we have an example of our ionic bonding. So as you can see, they all make really, really different shapes and really, really complex structures. And then that leads us on to the quaternary structure. Now, our quaternary structure doesn't always happen. So all proteins have a primary, secondary, and tertiary structure. Some stop at the tertiary structure. Some continue on to the quaternary structure. Now, the quaternary structure is made up of 2 or more polypeptide chains. So up until now, primary, secondary, tertiary structures was just one polypeptide chain, and the folding, because of the bonds and the interactions between the molecules and the R groups, and getting more and more complex. So we have this alpha helix, the beta pleated, and we're forming all these different disulfide, hydrogen ionic, and hydrophobic bonds. Now, quaternary structure, we're going to actually add in another polypeptide chain. Okay, so this is what makes it more complicated here. So we've actually got 2 or more polypeptide chains. So some proteins will stop at the tertiary structure. If we've got 2 or more polypeptide chains, then we have a quaternary structure. Hemoglobin is one example of a protein that has a quaternary structure. It is also an example of something that we'll look at later called a conjugated protein, because it has another chemical group associated with it. And so we will call these a prosthetic group. And in these situations, it's the heme group, the oxygen binding site that makes the hemoglobin. Again, this isn't something we're going to look into too much now, but it's sort of good to sort of have this idea of sort of knowing about it. Prosthetic groups are not going to really come into place in tertiary structures. It's only when we look at quaternary structures. So it's the iron that has the heme group in hemoglobin that is the prosthetic group in this situation. So our quaternary structures, well, what basically happens is we've got this bit here. Hemoglobin is actually made up of 4 different polypeptide chains, and you can sort of see that from this image here. We've got 2 dark blue ones and 2 light blue ones. That's our 4 polypeptide chains. 2 other examples of proteins that have a quaternary structure are things like collagen and insulin. Unfortunately, we then start to make proteins a little bit more complicated. We go, okay, there's a primary, secondary, tertiary, quaternary structure. Then we start to group them into globular and fibrous proteins. So proteins can then get divided into these 2 main groups. If we think back to secondary structure, that we have alpha helix and beta pleated sheets, you can sort of see that we're still sort of sticking more towards it. It might be more one or more the other. So globular proteins tend to be more spherical in shape and soluble in water. And so hemoglobin is an example of a globular protein. Why this happens is because they have a hydrophilic R groups on the outside of the molecule. Like we said before, hydrophilic R groups are going to be on the outside, hydrophobic R groups will be on the inside of the molecule. The specific shape of globular proteins is very important for their function. So examples are hemoglobin, enzymes, myoglobin, antibodies, and transport proteins. They are not going to want to be hydrophobic molecules. They are not going to want to be molecules that are not soluble in water. It's really, really important that these are soluble in water. They're really involved in really important metabolic reactions. And also the shape, not only is it important for their function on whether they're soluble or not, it's also actually really critical for their binding to other substances. So when we look in detail later at things like transport proteins and antibodies, they need to have that really specific shape. The antibodies need to have that specific shape, that specific antigen. If they don't have that specific shape, it's not going to work. So that's really important for globular proteins. So these are in a ball shape. So here's an example of globular protein with a quaternary structure, and that's hemoglobin. So similar to what we saw before, and you can see those prosthetic groups. Here they've used them as like an orange tic tac almost, being the heme group from iron. It's made up of 2 alpha subunits and 2 beta pleated sheet subunits. That's just something to sort of know. And it has those 4 heme prosthetic groups. Then we have our fibrous proteins. Fibrous proteins do not fold into a ball shape. They are a long chain, a long strand still. Because of this, they are usually insoluble in water. And they have very structural roles. As you start to think, okay, the long, the fibrous, they're insoluble. That's actually quite good if we're going to have a more structural role. So things like keratin in our hair and skin are fibrous proteins, tendons, cartilage, bones, blood vessels. And so we're going to start to have these sorts of shapes, double helix, triple helix shapes, make fibrils, and it's this structural, integral sort of shape that we're going in. So they have lots of cross links happening here. So as you can see, and in this one, where the collagen fibers are, they're cross linking across. This increases the strength. So fibrous proteins are really, really strong. Their shape is less important than the globular ones. The globular proteins have these really specific shapes that are really important for their function, using that lock and key enzyme theory. Whereas for fibrous proteins, actually, their long strands are more important because it's going to create the strength that's required. I have no clue what that says on the top. Let me just move this. So still fibrous proteins. I think we're nearly at the end. So fibrous proteins, long chains, stuff for 4 structures. They're insoluble in water. And so we get this shape again. So skin, teeth, bones, and we have these cross-linking parts that make them really, really strong. So they can make a triple helix of a polypeptide chain. And these chains are held together by those hydrogen bonds, and they are really, really, really super strong. Transport proteins are going to usually be globular. Cell signaling, again, going to be globular. Enzymes, globular. Antibodies, globular. Structural, fibrous. That's sort of how it works, just because of the basis and how they fold. When we next come back to proteins, we'll look at this actually in a second, we will then look in a bit more detail at enzymes and DNA, so sort of looking at specific examples of proteins because we have to know them in a bit more detail. So yeah, proteins, we do need to know how to test for proteins. Hasn't changed since GCSE, which is really quite nice. Again, you're going to place your food sample in a test tube. You're going to add that Biret reagent. Sometimes it comes as birette A, birette B, birette 1, birette 2. You have to mix the two solutions together. You need to mix the same quantities of both solutions together. And birette reagent is alkaline copper sulfate. And if a protein is present, the solution turns a lilac colour. This is also one that as you can see, we've got water, albumin, starch here. The albumin has got protein in it. And so it will just turn from a blue to a purple colour. I think, yeah, that is it. So let me stop recording.